In this video, we're going to take a look at enzymes and how they are regulated. This is a second video on enzymes. So when we're talking about anabolic reactions, there has to be an input of energy because the product is more complex than the reactant. So if we look at this example here, we have the enzyme with the open active sites. The two individual substrates bind to the active sites, releasing of water because it is most often a condensation reaction. A new bond is formed between the initial substrates, creating a bond. There had to be an input of energy to create that bond and to stabilize it. Noticing that the enzyme is released as it initially was and can be reused again. So there are several steps to make this happen. So the first step is that the substrate has to bind to the active site. More often than not, induced fit occurs, meaning that the enzyme changes its shape to embrace that substrate or to have a better hold on it. The substrates are held into that active site by weak interactions, most commonly hydrogen bonds or ionic bonds between charged entities. When they are being held in that active site, the enzyme then lowers the activation energy and speeds up the reaction. As we learned in the previous video, we can remember it by the MODS squad. So there's a creation of a microenvironment, there is proper orientation, there is direct interaction, or there is the straining of the bonds. What will then happen is the substrates are converted into the final products with new bonds being formed. The products are released and the enzyme activation site is then available to repeat the process. A common example of an anabolic reaction using an enzyme is the creation of ATP from ADP. Notice we have adenosine diphosphate or ADP binding to the enzyme here. We have the inorganic free phosphate binding to the enzyme as well at the active site. The enzyme then allows those phosphates to create a new bond, releasing the products. Noticing when we write the reaction, we write the enzyme, in this case ATP synthase, over top of the arrow because it is not consumed or used up as part of the reaction. In catabolic reactions, we have the overall release of energy, and this is because we end up with less complex products than we began with. So in this case, we have a disaccharide binding to an enzyme. The enzyme will then lower the activation energy. In this case, it is a hydrolysis reaction, so water is added to help break that bond, and you have two monomers released. The enzyme can be recycled again. So as we went through the steps in the anabolic, let's go through the steps on catabolic. So the enzyme and substrate, both available and can bond to one another. The substrate binds to the enzyme. So this means that it has to come in contact in the proper orientation to actually be able to fit to the active site. It can't bind anywhere else. Substrates are then converted into the products. In this case, it is a hydrolysis reaction, so water is added to break that bond. The products are released and the enzyme can be recycled. An example of this would be ATP reverting back into ADP and an inorganic phosphate. The enzyme used for this is ATPase. So there are factors that affect how well an enzyme can work. Some things will improve their ability to work and some will decrease. So the first one we're going to look at is substrate concentration. So our substrates are the molecules that are going to bind to the active site and that are either going to be made more complex or less complex by the enzyme activity. So when we have very few substrates, we have very low substrate concentration. When we have many substrates, you would assume that the reaction is going to happen a little bit faster because it's more likely that that substrate will come in contact with the activation energy. And that is the case. So if we look at our rate of reaction or the speed at which the reaction occurs, and then increasing substrate concentration along that x-axis, notice that the reaction will increase steadily until we get to this point here. This point is what is known as the point of saturation, meaning that if we increase the amount of substrates in the environment, we are not going to make that reaction happen any faster because those enzymes are all full at any given time, even though there are still more substrates that could possibly bind. So we have this point of saturation when we're talking about substrate concentration. Enzymes also have optimal temperatures and pH levels. So if we take a look at optimal temperature, noticing that as we increase the temperature, there is a more rate of reaction, or it happens faster and faster. Well, if you think about particle theory, the more that those molecules are moving around, the more likely they are to bump into one another. Therefore, the more likely the substrate will find the active site, more likely the reaction will progress. However, we do have what is known as an optimal temperature. Because if we go too much past that, or if we get warmer, well, most enzymes are proteins. Proteins, when they get too warm, will denature, lose their shape. Therefore, they no longer have an appropriate active site. So you see the rate of reaction drastically decrease after that optimal temperature because we are beginning to denature or change the shape of that enzyme. So some examples of that is we can see that different enzymes are going to have different temperatures. They don't all have the same temperature. So a human example of that is men's testicles are outside of the body because the enzymes to create sperm work at a slightly lower temperature than internal body temperature. So this having them outside of the body allows it to be slightly cooler. So different enzymes will have different ideal temperatures. pH level also affects enzyme activity. So if we think about enzymes being proteins and how proteins get their tertiary and quaternary structure, pH will change the environment, which will change how those proteins fold and take their shape. So enzymes will have an optimal pH. If you become more acidic or more basic, their shape will begin to change and they will no longer be as effective or effective at all. So some examples of this are pepsin, salivary amylase, and arginase. Pepsin is used to break down proteins in the stomach, and we know that stomach acid is highly acidic, so noticing its optimal pH is very acidic. Salivary amylase is used in the mouth, in the saliva, to primarily start breaking down carbohydrates. So our mouth is not as acidic as our stomach, so its optimal pH is somewhat near neutral. Whereas arginase is used in the conversion of urea to make it less poisonous in the liver tends to have a more basic environment, so it has a different optimal pH as well. This also allows for enzymes to be turned on and off as they progress through. So as the food moves from the mouth to the stomach, salivary amylase will become denatured and will no longer continue breaking down those molecules once they have reached the stomach. There are also what are known as enzyme cofactors, and these activate enzymes or proteins. So some enzymes need them to function. So if we look at the inactive enzyme or protein, its active site isn't quite the right shape. So when a cofactor binds to it, it will activate it, meaning that its active site is now the appropriate shape. Most commonly, these are metal ions that are found in foods that are consumed. There are also what are known coenzymes. These are organic cofactors, or substances that are made by organisms. They most commonly transfer energy in the form of electrons and allow bonds to form or break by transferring that energy. Common examples are many of the vitamins and NAD+, which we will see later when we get into cellular respiration. There's also what is known as allosteric regulation. So this means that cells can control enzyme activity in order to coordinate, because cells don't want to use up energy if they don't need it. So they don't want to keep producing a molecule if it doesn't need to be produced, because it always takes an input of energy to get those reactions going. So when we look at an enzyme, there's what is called the allosteric regulatory site. It is different than the active site. It's another location on the enzyme. So when a substance binds to an allosteric site, it can cause a conformational change in the enzyme, either activating it or deactivating it, so it can or cannot bind to the substrate. So if we look at an allosteric activator, it means that a molecule will bind to the allosteric site and will change the shape of the enzyme such that the active site will fit the substrate. Whereas an allosteric inhibitor, the inhibitor will bind to the allosteric site, and this changes the active site shape so that the substrate can no longer bind. This is known as a non-competitive inhibitor because it is not competing at that active site with the substrate. However, there are competitive inhibitors, meaning that they have a somewhat similar shape to the substrate, and they can fit into part of that active site and actually block the substrate from binding. So when we see here the substrate and the enzyme, competitive inhibitor binds or blocks the substrate directly from getting to the active site, whereas a non-competitive inhibitor will bind elsewhere on the enzyme, changing the shape, making it so that the substrate can no longer bind. The last type of regulation we're going to look at is feedback inhibition. Now, this is when there are a series of different enzyme catalyzed reactions, so such as cellular respiration. It's not just one reaction and everything happens. It takes several steps, and each step requires an enzyme to make it happen. What happens is the product from one reaction becomes the substrate from the next, and the product becomes the substrate. And at some point, one of those products will bind to a previous enzyme in that chain reaction. And by binding to that previous enzyme, it will inhibit it. So it could be competitive or non-competitive inhibition, but it prevents a previous substrate from binding to an enzyme and shuts the entire process down. So one example of this is the creation of the amino acid isoleucine. So there is an initial substrate that is enzyme catalyzed through several different steps, and at the end, isoleucine is created. Well, if your body or cells have enough isoleucine, it's not worth putting in more energy to make more. It's a waste of energy. So that isoleucine comes back to the initial enzyme, binds to it, changes the shape of the active site, so the initial substrate can no longer bind. However, as the body uses up the isoleucine, it will eventually use this one, pull it off the enzyme, the active site now becomes available, and the process can continue. ATP is regulated like this in the body as well because the body can't store ATP, so you don't want to make it if you don't need it.